Okay, so now that we saw the different parts of the cells, we're going to move into the functioning of certain cells that works with electrical signals. So when these cells that works with electrical signals, which are like the cardiac muscle cells, smooth muscle cells, neurons, when they are resting, these cells, they have internally an electrical charge, a voltage, that we call resting membrane potential, or RMP. So this resting membrane potential, or this voltage that these cells have during resting, it happens because they have internally in the cytoplasm, these cells, a specific amount of ions. Sodium, potassium ions are the major contributors for this resting membrane potential. And with this permeability that exists in the cell membrane, these ions can be exchanged and the voltage can change too. So resting membrane potential can be defined as the difference of voltage across the plasma membrane at rest, which in most cells is minus 70 millivolts. There will be some that they would have a little bit less or more voltage depending on the cells, but it's always negative, the resting membrane potential. And then something that helps to maintain this specific concentration of ions or gradient across the plasma membrane, it is the sodium-potassium ATP pump. So ATPase pump. So this pump uses ATP in order to exchange sodium for potassium so that you have a specific ratio or concentration of potassium and sodium in and out of the cells. So don't think like the cell has only, for instance, potassium ions in the cytoplasm because it's the major cation that it is found in the cytoplasm, but also outside of the cells there is potassium. Like if we were in a resting membrane potential and if we were all potassium ions and this room was the cell and this is the cytoplasm, well, we are highly concentrated right now because in the hallway there's no one, and the hallway will represent the external environment where outside of the cell. So this room is the cell, we are the ions, the potassium ions. But just because we are concentrated a lot in here, all the students, that doesn't mean that in the hallway there's no students. So there is students outside the hall and also here, but they are less concentrated than you guys here. So that's what happens with this. So then here is how these ions maintain the gradients, the resting membrane potential, how they maintain the resting membrane potential. So sodium, which is positively charged. So remember, cations have a positive charge. So these are ions that has a positive charge, while anions have a negative charge. So then because sodium, this cation, is more concentrated out than in, and potassium is more concentrated in than out, that's what it generates this electrical potential. And you might say, well, both are cations, why the cell is not positively charged? Well, it's the ratio And as well is the presence of anions within the cell, which are anions coming from the phosphate groups, from the phospholipids, phosphate groups from amino acids, from proteins, and phosphate groups that are attached to molecules like ATP, like DNA. So all of these negative charges favors the cell to be more negative than positive at rest. There will be a change when the cells become active where these negative charges reverse. So instead of being negative, you have positive charges inside the cell. So now we have this pump, the sodium potassium ATPase pump, that moves 3 potassium out and 2 potassium in, so that you maintain that ratio. So you have then more potassium inside and sodium outside because of that ratio given by this sodium potassium pump. And here is how the sodium potassium pump works. So it's a carrier, and it's an antiporter. Antiporter will be basically exchanging. So things can go in and out of the cell at the same time. Those are antiporters. Symporters can bring 2 molecules at one inside, and uniporters just 1 molecule inside or outside, depending. So this is a sodium potassium pump, and it has 3 little gaps that fits sodium, while it has 2 little gaps that fits potassium. So once it gets potassium charged or loaded, ATP is used and it's converted, it's broken down into adenosine diphosphate and inorganic phosphate. And that's what's driving the opening of this part of the channel. So see how this part of the channel is closed initially? But once you bind ATP, you have a configuration change, so the shape changes. So now this part opens, and then since you have loaded already your sodiums here, this sodium is ready to go out. And then you will have, as this sodium is getting out, potassium coming in, and then this part closes, and now potassium is available to be discharged to the inside of the cells. So that's how it works, and you require ATP. More or less, you saw this before when I was explaining the parts of the cell in the previous lecture. But organelles are defined as specialized cell structures that perform specific essential functions. So we have organelles that make the ATP for the cells, like the mitochondria. The mitochondria has a double membrane. The inner membrane makes foldings internally, and these foldings are called cristae. And inside these foldings, we have a matrix where you have the enzymes that are going to generate part of the molecules that are going to help us to make ATP during the Krebs cycle or tricarboxylic acid cycle. And then between these two membranes, we have molecules that act as carriers of hydrogen, and that helps for transfer electrons along this chain of molecules, and that will help to generate most of the ATP of our cells. So mitochondria is the powerhouse of the cells. And an example of what happens when mitochondria is damaged. So if mitochondria is damaged, then the cell won't produce as much ATP as needed. So, clinically speaking, someone who has a mitochondrial disease is more than likely to have fatigue because he won't have ATP to drive the metabolic reactions. Now, our mitochondria come from our maternal side. So our mom gives us the mitochondria that we have. So if someone has a mitochondrial disease, that disease is inherited through the mother's side, not through the dad's. Now, it's not an organelle, but it is found in the cytoplasm, ribosomes. Ribosomes makes proteins, and it's made out of RNA and protein complexes. They are forming the nucleus initially in the nucleolus, and then they exit the nucleus, and then they can be free-floating in the cytoplasm, or they can be attached to the rough endoplasmic reticulum. So in that case, you will have an RER, rough ER. Function is to produce proteins by translating messenger RNA. So messenger RNA carries information from the DNA into the cytoplasm so that you can start adding amino acids like the monomers of proteins so you can make a protein. And that code of the DNA tells us specifically what amino acid is supposed to be there. We have 20 types of amino acids, and out of these 20 types, we have all the different proteins that we have. Examples of this dysfunction will be defects that can impair protein production, so you don't produce as many proteins, and that can contribute to diseases like Diamond-Blackfan anemia. Endoplasmic reticulum. It's a network of tubules made of plasma a membrane, a lipid membrane. And the one that has ribosomes is going to be called rough ER, while the one that doesn't have ribosomes is going to be the smooth ER. And the rough endoplasmic reticulum, because it has these ribosomes, it will be making proteins, while the other one, the smooth, makes esteroids, lipids, and detoxifies the cells. So something that you can expect then is that if a cell starts making a lot of proteins, it's going to increase the size of the rough ER. And now, if a cell is detoxifying, for instance, a lot of alcohol, then the cell will have a lot of smooth ER. And the area of the body or the organ of the body that we use to detoxify alcohol is the liver. So if someone drinks a lot, then it will have a lot of smooth ER in the liver cells. And the liver cells themselves, they will have a lot of smooth ER. So examples of endoplasmic reticulum stress. Well, it can lead to neurodegenerative diseases and diabetes. Golgi apparatus. In Golgi, we, similarly as for rough ER, we have tubules of membranes that actually are called cisternae or sacs. And this one is the center where the recently translated proteins from the rough ER goes and be sorted out to know where they're supposed to go. So they are packed and they're tagged so that they can go into a particular area. Like, what if an area of the cell needs to have sodium channels? Well, in that case, if the messenger RNA is giving the code for making a sodium channel, and that sodium channel is made, then you have to bring it into that area where it's needed. It's like the UPS system of the cells, the Golgi. Dysfunction of Golgi can lead to congenital disorders of glycosylation affecting multiple organs. Now, lysosomes are small vacuoles, like little pouches or sacs of plasma membrane, where you can have digestive enzymes, and lysosomes are produced by the Golgi. Function is with these enzymes, the lysosomes can break down waste and recycle cellular components. So many times the cells, they need to be repaired because one of the organelles may get damaged. So in this case, the lysosomes can fuse with these damaged organelles, and then you can start breaking down this damaged organelle, and then you recycle the components. So, example of lysosomal storage diseases, we have Tay-Sachs, and that results from enzyme deficiencies that causes toxic buildup that affects the neural cells. So another view of these organelles. This is the nucleolus. The nucleolus is inside the nucleus, so all of this is the nucleus. In the nucleus you have the nucleoplasm, which is analog to this cytoplasm, and that's where you have the chromatin, the DNA. And then in here, just next to the nucleus, we have the rough endoplasmic reticulum with these ribosomes attached to them. This is the smooth endoplasmic reticulum. Here's the Golgi apparatus releasing vesicles for exporting proteins, for instance. Here we have the mitochondria. Here we have some lysosomes, peroxisomes, centrioles, the organizing center of these microtubules, that these microtubules bind the chromosomes when they are about to divide in mitosis, when the cell is going to separate these chromosomes. And then in here, we have cytoskeletal proteins, and all of these will be the plasma membrane. So protein synthesis, it's given at the ribosomes, or is made at the ribosomes, whether they are free or in the rough endoplasmic reticulum. And we have a genetic code that gives instructions about what amino acids will go in that protein and in what order. And this combination of amino acids in that particular order, and even in particular numbers of amino acids, is going to influence the secondary shape, whether you have an alpha helix, a beta pleat, or or that tertiary structure, like how this protein will fold, the three-dimensional shape that it will have. Because these amino acids have charges, and these charges can be used to have hydrophilic, hydrophobic interactions, create bridges between amino acids with hydrogen bonding, etc. So first, what you need to do is get a transcript out of the DNA. That transcript is going to be called messenger RNA. So that's transcription. So you copy part of the DNA, make messenger RNA, and then the messenger RNA exits the cells and the nucleus, cell nucleus, and then you will bring this messenger RNA into the ribosome so that they can be translated, so you can make your protein. And then the messenger RNA is going to interact with the transfer RNA and with the ribosomal RNA so that you can make a protein. So all three RNAs are required to make a protein: messenger, ribosomal, and transfer RNA. So here is the genetic code. So these are read in triplets or in groups of 3. So these uracils will be coding for phenylalanine, these 3 uracils. So if you have uracil plus a cytosine, you form phenylalanine. So here is the first base. So you have U in here, C in here, A in here. Uracil, cytosine, adenine, and guanine. This is RNA. RNA is characterized by having uracils instead of thymine. Thymine is found in DNA. And then this is the second base. So everything here, the second base is going to have uracil because you combine this with whatever letter is in here, whatever base is in here. And then for the third base, here we have UCAG, so here will be UCAG, UCAG, UCAG. And each of these different combinations of the triplets will tell you what type of amino acid you will bring. And then here's the abbreviation for the amino acids. So alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, etc. are the different amino acids that we have. And here we have, for instance, another example. If you have 3 Gs, that codes for glycine. If you have 2 Gs and 1 A, that codes for glycine. Same for 2 Gs and Cs, glycine, 2 Gs and Us, it's a glycine. So this code is what they call degenerate, but this one help us to have some sort of flexibility when it comes to the third base, which is called the wobble base, because in this case, with changes in this third base, you can have different types of amino acids. Like, for instance, here you have AUU, which is serine, but if you have AGA, that's arginine. And now, every transcript is going to start with AUG. That's the start. That codes for methionine, or the start. And when you reach the end of the transcript is going to have stop codons. So all of these triplets are called codons. So UAA, it's a stop codon, UAG is a stop codon, and UGA, it's a stop codon. Now, let's say that one of the amino acids in the middle of your transcript has this code, UAC, that it's supposed to be tyrosine. But then you have a change in the DNA, a single point mutation, that instead of having a guanine, that will be the one that it's supposed to base pair with cytosine, or that code, sorry, for the transcript for cytosine, instead of a guanine, you change it for a thymine. In this case, your codon is going to change from UAC to UAA, and that will lead to a stop codon in the middle of the protein. So you end up with a protein that is not complete. So sometimes these mutations can lead to problems because maybe you will by needing this protein, like if it was an enzyme to perform a metabolic reaction, but you cut it in half. Basically, you don't have a complete protein. In this case, the metabolism of that particular pathway is gone. So some of these mutations are important. A single amino acid can change the outcome. So then, with this AUG, the start codon, you will start bringing your first amino acid. So in this case, it's going to be methionine. What you can see here, it's an open view of a ribosome. In the ribosome, you have a little gap through which the messenger RNA transcript goes in this direction, and then you start reading with this transfer RNA, this one that is carrying this amino acid, this MET, is methionine. This is brought by this transfer RNA, and as this messenger RNA is elongated, so this is initiation, you bring more amino acids. So in this case, the next one, since it's going to be GCC, now it's going to be glycine. And the next one is going to be glycine as well. So you keep bringing them, and once you have your first amino acid created or brought by this transfer RNA, you form a dipeptide bond with the second amino acid, and then your first amino acid is free to go. No, you release it. And then you bring another type of amino acid, and then you keep elongating until you have your complete polypeptide. And then once you reach your stop codon, which in this case is UAG, you bring something that is called a release factor that is not going to have any amino acids. So then you stop making your proteins. So all that it takes is grab a piece of this code from the DNA, make a messenger RNA. The messenger RNA, it can be partially processed in the nucleus before it exits. So in the DNA, we have introns and exons. So you remove introns, you leave the exons, and then you can recombine some of these exons, and then you can make different proteins. And once you have whatever final version of the messenger RNA needs to go out, exit through these nuclear pores, then goes into these ribosomes, and then you bring the transfer RNA, you bring your amino acids, you elongate your chain of amino acids, create your dipeptide bonds, and then once you reach the stop codon, you stop producing your protein, and then you give a three-dimensional shape. So this is the secondary shape, and then you give the three-dimensional shape, which will be in some cases tertiary, and in some cases quaternary for some proteins. So that's protein synthesis from transcription to translation. Any questions? Okay. So now let's look at what is cellular respiration. So cellular respiration is basically converting glucose into ATP, which is what our cells uses as a source of energy. So in cellular respiration, you have several processes that lead to the formation of ATP, which includes glycolysis, which is breaking down glucose, happens in the cytoplasm. Krebs cycle, which is converting pyruvic acid into different components of the Krebs cycle. And this happens in the mitochondria, the Krebs cycle. And then you have the electron transport chain happens in the mitochondria into where you produce most of the ATP, and as a byproduct of cellular metabolism, when you're producing ATP, you produce also CO2 and water. Water is very few the amount that you produce, but CO2, it's a toxic waste, carbon dioxide, so we need to get rid of it. And of course, since you generate ATP, that gives you energy. So glycolysis, Krebs cycle, and electron transport chain forms cellular respiration. So now let's look at what are the phases of the cell cycle and the types of cell division. So we have a cell cycle that has two major phases. It has inner phase, which will be all this, and it's called a cycle because it repeats. So it goes like this. Right? So this is the beginning of inner phase, this is the end of inner phase, and then you have another phase that is called mitosis. Major events happens during both. So inner phase is when you duplicate DNA and organelles in preparation for mitosis. So this will be, let me put the title here, cell cycle. And then in here we have mitosis. So mitosis is subdivided into 4 phases. So the first one is going to be prophase. The next one is going to be anaphase. No, sorry, metaphase. Anaphase and telophase. So in mitosis, the DNA that you duplicate here, you will divide it into 2 equally into 2 daughter cells. Now, your inner phase is subdivided into G1, so this is G1, into S phase, this is S phase, and into G2. And some cells, they enter a phase, not all of them, that is called G0. So cells that won't divide again, once they divide, they enter G0. Like neurons, like muscle cells. And specifically in S phase is where you have DNA synthesis. That's when you duplicate your DNA. And then in between here and here, like here in G1, that's when you grab all the materials that are necessary for duplicating the DNA and making more organelles. And then in here, you duplicate your DNA once you pass this G1 phase. And then in G2, you just make sure that you have DNA without major problems, and that you have equal number of organelles as well. So we have then G2 as preparation for mitosis, and M phase or mitosis includes cytokinesis, which is when the cytoplasm of the cell is going to split in two. So in prophase, your nuclear membrane starts disappearing, and then you have visible chromosomes. In metaphase, your chromosomes align in the center of the cells. So this is metaphase plate. Oh, let me put here. And then you have your centrioles here. So these are the centrioles. So your centrioles in metaphase are at different poles. And this is another centriole. And you start forming like a rope that is going to attach these chromosomes. So this is the mitotic spindle. And then this area of the cell in the middle is called the equator, just like the Earth's equator. It's a horizontal line across the globe. And then the next phase, so let me underline each phase. So the next phase is going to be called anaphase. In this case, in anaphase, you start pulling apart your chromosomes, your sister chromatids. So the chromosomes have sister chromatids. It's like little twins. So they are seen farther apart. And then in telophase, you have more apart these chromosomes. And then you start forming again the nuclear membrane. And then you have a little indentation in the cell that is going to be called the cleavage furrow. So as telophase is going from beginning to end, this cleavage furrow will become deeper, and then you separate the cells, and then you have two daughter cells. Hopefully with equal number of chromosomes, no mutations, and equal number of organelles. So parents who have only one child, giving whatever things that they like to give to the child, doesn't matter if it's too much or too little, because it doesn't have someone to compare to. But when you have siblings, especially there are two, and they are kind of close in age, or maybe not even close in age, but eventually the younger for as much young as is as the older one. If you give more to the older than to the younger or vice versa, there's going to be problems. So better be fair. So same with the daughter cells. You give equal number of chromosomes, equal number of organelles. Now for meiosis. Meiosis is basically two sequential cell divisions that are required to make gametes, which are sperm cells and egg cells. And we have in mitosis this process to repair tissues or to make organs to grow or tissues to grow. And in mitosis, the DNA of the daughter cells has to be identical. In meiosis, it cannot be identical. Why? Because otherwise, we won't be who we are, which we are unique. Each of us is unique. Yes, we have similarities with our siblings. Some of them are maybe better looking than you, according to them, but that's not true. You're always the better looking one. That's a fight, always, always. It's a fight with the siblings. Who's the smartest, who's the best looking?
You ever heard identical twins argue?
Even with the identical twins, you are always the best.
I'm an identical twin, we argue.
Huh?
I'm an identical twin, we always argue, like, who's uglier, we look exactly the same.
But you're the handsomer, right?
I think so.
Yes, I think so too. Then when I meet your twin. No, it's not true. No, I'm kidding.
One of these days we'll switch places to see if we can figure it out. I'll be in Embassy, let's go come to Weston.
Don't switch places in the text. Don't do that. All right, good to know. But then, even with identical twins, which are actually the two human beings that are the closest in DNA similarity, there are differences. And it's related to this basically genetic variation that comes from meiosis. So, this is a better view of what I drew there. And so cells spend most of the time in interphase. And then which is all this. And then you have then again G1, which is where your cells grow. And S phase when your duplication of DNA takes place, and G2 where you have further growth in preparation for cell division, and then you enter the mitosis, which is very short period of the cell cycle, and then cytokinesis where you split the DNA. So basically mitosis is the splitting of the chromosomes in the nucleus, or, well, separation. And then your daughter cells will have to be identical. And again, some cells opt to not divide again. And that's kind of like the precious cells that we have in our body, like the neurons, heart cells, etc. So then the difference, again, between mitosis and meiosis. Mitosis produce identical cells. Meiosis produce gametes with half of the chromosome number. In mitosis, you're supposed to have equal the chromosomes in number as well. Sometimes something happened with this spindle apparatus where you don't drag these chromosomes apart, and then instead of carrying one to each daughter cell, maybe this cell on the top is going to receive one of these pairs of chromosomes, is not going to receive anything, and this one is going to receive two. And that's how we end up with trisomies, like 21 chromosome trisomy, which is known as Down syndrome. This is the most common one because this is like the one that survives the most. But we have other ones like Edwards syndrome. We have trisomies for 17, for 18, and they die at very young age. And some trisomies, they don't make it. I mean, they can be aborted. So it's something that it can happen. So this is the microscopic view of these phases. So this is inner phase. In inner phase, your chromosomes are not visible. This is the nucleus of the cell. This is the cytoplasm. This is the plasma membrane. In prophase, these chromosomes start condensing, so you have them visible. Chromosomes have arms. These are the arms of the chromosomes. They have a central part that is called the centromere. And then in prophase, not only your chromosomes become visible, but your centrioles, which are these, start moving apart and then start attaching microtubules to form the mitotic spindle, and your your nuclear membrane starts disappearing. It has to disappear. Why? Because the microtubules, they have to connect to the center of the chromosomes, the kinetochores. And this is the metaphase plate. This is metaphase. So you have here your centrioles, your spindle fibers, which are these, and these will be the chromosomes aligned in the center. So that's metaphase. And then in anaphase, you start pulling apart the chromosomes by shortening these microtubules. And then in telophase, the chromosomes are definitely farther apart, and then you start forming again your nuclear membrane. And then you start forming this cleavage furrow, and then in cytokinesis, where your cleavage furrow goes all the way, you separate your two daughter cells. So this is how it looks in the microscope. And then again, sometimes these fail to separate, so you end up with non-disjunction and a trisomy. So what is the difference between chromatin, chromosomes, and chromatids? Chromatin is the DNA plus the histone proteins, and this chromatin is loose during interphase. chromosomes are condensed chromatin that is visible during mitosis, and the chromatids are basically half of these chromosomes, a replicated chromosome. So you have two sister chromatids in each chromosome, and then you pull them apart during mitosis. So let me, before I continue with that, let me write you down the number of chromosomes that we have in the somatic cells. Diploid cells have two sets. This is 2n to the number, two sets, or 23 pairs, which is 46 chromosomes. And then sex cells, which are the sperm and the egg have 23 individual chromosomes, 1n, one set, or haploid. Now for DNA replication. So you have to split or separate these strands of DNA. If you remember, DNA has two strands that intertwine like a helix, and then here you have your nucleotides. So in order to expose these nucleotides, you have to separate them with an enzyme that is called helicase. And then when you have exposed these nucleotides, then you can make a copy and you bring an enzyme that is called the DNA polymerase. But you add first these primers of RNA, which corresponds to the complementary of whatever code you have in your DNA. In this case, this is AGC. So cytosine would have to base pair with guanine with cytosine, and adenine with uracil, because this is RNA. So this is an RNA primer. And then that enzyme that makes these primers is called the primase. And then as you open with this ligase in this direction, the double helix, this primase, the DNA polymerase can start making a copy. Or you can transcribe it with an RNA polymerase, depending on what you're doing, DNA replication or transcription. If it's transcription, it's going to be with uracils, like in this case. But if it's a DNA copy, it has to be with thymines. So And then you have 2 new copies of DNA in this case. And then once you make the copies, you will have to wrap it around these proteins that are called histone proteins that has a positive charge. Since the phosphate groups of negative charge in the DNA get attracted to this positive amino acid that the histone have, it's easier to give 2.5 turns or so and then link them. And then you can wind up all of these, and then you end up with a chromosome that looks like this. This is 14 nanometers, so it's a very small unit for measuring microscopic structures. And this is the central part of the chromosome that is called the centromere. This is the short arm, this is the long arm. Short arm is also known as P arm, and the long arm as Q arm. And these are sister chromatids. So you have 2 sister chromatids in a duplicated chromosome. And the endings of the chromosome are known as telomeres. These telomeres shorten as we age. We have an enzyme that is called telomerase that is supposed to elongate the tip of the arms, which is the telomere, but it seems like that is related to aging, the lack of elongation. And then this is how a human chromosome look like. The genome looks like in a photograph that is called the karyotype. So here is pair number 1, 2, 3. So we have 22 somatic autosomal pairs and 1 sex pair. So they can be stained, so they can be extracted from the nuclear envelope or the nucleus. They can be stained. You can take a photograph, and then you can pair them. And the longest is going to be the first pair. The shortest is going to be the 22nd pair. And then this individual has an X and a Y. The absence of a Y is what it determines gender. So sometimes in these problems of bringing the chromosomes into the cells and out in mitosis or meiosis, you cannot bring, for instance, one of the X chromosomes into one of the egg cells or the sperm cells. And then the egg cell is still fertilized, but is lacking 1 X chromosome. So in this case, it's going to have only 1 X. And that individual is going to be a female. And that is called Turner syndrome. So that's a monosomy. In this case, you have 1 member of the pair. And then if you have 3 members of 21, that would be Down syndrome. So then this is how the karyotype look like. And then we already saw this genetic code, how this DNA codes for that. Any questions? Okay, so for histology, basically you're going to understand in this chapter what are the different types of cells that we have, how do they form tissues, and what are the four main type of tissues that we have, and how they start forming organs. Test your knowledge. If a disease affects the mitochondria, which cell function will be lost? Or what function will be lost? A, B, C, or D? B, ATP production. Number 2, what type of membrane transport requires ATP to move substances against their gradient? Simple diffusion, facilitated diffusion, primary active transport, or osmosis? From less to more, what do you need? Simple, facilitated, primary active transport, or osmosis? Osmosis is the movement of water from more concentration of water to less concentration of water. So it's not osmosis. A, B, or C? C. Mhm. True or false? The cytosol includes all the organelles within the cell. False. So the cytoplasm is the one that actually includes all the organelles within the cell. Cytosol is the liquid portion of the cytoplasm. True or false? Osmosis is the movement of solids from low to high concentration. False. Why? It applies to water, not to solids. True or false? The sodium potassium pumps move 3 potassium out of the cell and 2 sodium in. False. Is 3 sodiums out and 2 potassium in. Short answer, briefly explain how selective permeability of the plasma membrane supports cell homeostasis. Can everything come in and out of the cell freely? No. If that will happen, then we will have electrically excitable cells. So the neurons won't function. So that's why you have to maintain permeability, selective permeability.